Well, I guess it's a pleasure today to welcome Professor Satoshi Yoshiji
Yes.
to the QLS seminar series. He is an assistant professor in the Department of Human Genetics, part of the McGill Genomic Circ program at the McGill Genome Center. He's affiliated with Quantitative Life Sciences, also one of the newer arrivals in Quantitative Life Sciences. He's an endocrinologist by training. And then after he did one of the Kyoto McGill programs, and then went to the Broad Institute for a while as well. I had the pleasure of working with him a few years ago during his PhD. So I'm looking forward to hearing your work today, Satoshi.
Thank you, Cynthia. And thank you so much for inviting me. It's my great honor to be back here. I think my last time here is TNU1's thesis defense. So yeah, it's great to see, people are keep joining QLS. And I think this is one of the most amazing programs at McGill. There are a good number of people who understand life science, but there are a good number of people who understand quantitative analysis, but there are not many people who overlap. So I think this is one of the great programs, and I'm so grateful to be back here as a speaker. Okay, so let's get started. So please feel free to interrupt me. I have a bunch of slides, but I can't stop anywhere, so please. Okay, so my title is to leverage genomics, proteomics, and other omics for drug target discovery, especially for cardiometabolic diseases, with an ultimate goal of improving clinical care. Okay, so this is agenda. Who I am. So far here. Okay. So I'm a clinician. I have 6 years of clinical training through residency and fellowship back in Japan, and I'm board certified in endocrinology and internal medicine. And I have seen so many patients suffering from diabetes and its complications. Actually, I had to do ER shift 24 hours. It was a nightmare. But that gave me opportunities to see actually what's happening to patients. So patients, think diabetes very lightly in the beginning because they don't have a symptom. But when they realize, their kidney is so bad, they're on diet, they have to be put on dialysis, or they're like hard coronary arteries are so blocked. So even if you unblock one artery, next day the next artery can be blocked. So it's like a domino. So it keeps falling, but patient notice the seriousness until they do not notice it when you reach the very end. So my goal as an endocrinologist is to stop this domino in the very early part. So that's why I care about why patients are suffering, where treatments are lacking, and why more research are needed. But how we address this, or how I address this, that's why I decided to become a human geneticist. I did a PhD in human genetics at McGill. My mentor is Bram Richards, who is an amazing mentor. And I also did a postdoc at the Broad and Harvard Medical School with Dr. Jason Flank. He has a computer science background, and he is creating a common metabolic knowledge portal, which is like a gigantic platform to share the GWAS and other exciting science. And so my approach to science is ask clinically relevant questions based on my clinical expertise and address them with human genetics. So let's move on to research. So drug targets, this drug development is such a risky and costly business. So this is a recent article which came out in JAMA Network Open saying that the cost of developing one cardiovascular drug is like 890 million. Given like all the potential failure costs, like investments, taking everything into account, it's gonna cost $890 million. So if I do a startup, I don't think I'm gonna go into a cardiovascular medicine development because it's such a risky business. But there are fortunately some pharma doing this, but it's the same, $890 is a pain. And how can we address that? So human genetics can support the drug discovery. So there's a very famous article saying that human genetics evidence support around 66% of the 2021 FDA approved drug. And there is a follow-up study saying that when you take a look at drug which were approved or approved by FDA in the past 10 years, around like 70% have some kind of human genetics evidence. So human genetics help. So that's why my goal is to leverage human genetics for drug target discovery. So this is a topic I like to cover. The first is how we integrate omics analysis to identify causal mediators. And then I'm going to talk about a project with Chen-Yang Su, who is an amazing PhD student here, about creating a multi-ancestry atlas of protein disease associations. And then I'd like to cover about a new multi-omics and multi-ancestry biobank called BioPortal, for which I'm a co-PI. And lastly, I'm going to cover about the disentangling, the genetic heterogeneity of type 2 diabetes, which is a very intensely discussed topic. Okay, so let me start with the first one. So for the past few years, I have been very much interested in identifying circulating protein linking obesity to COVID-19 and cardiometabolic diseases. Why COVID-19? Because I treated patients who suffered from COVID-19. During the pandemic, I was still in Japan, taking care of severe COVID patient. It was such a hard time. And from the very beginning of the pandemic, it was very clear to me, people with obesity have very high risk of suffering from severe COVID. And I wondered why. And at that time, there was a huge development in high school proteomics and genomics. And there was a study using these proteogenomics data sets to identify proteins that are causal for COVID or other diseases. So I thought, okay, we can use this method to identify causal mediators. So given the time, I will not cover this topic, but that was published in Nature Metabolism. And I apply this framework to ask the next question, which is, okay, how circulating proteins mediate the effect of obesity on cardiometabolic diseases? And that is now accepted in Nature Genetics. Okay, so I'm gonna focus on this one, the cardiometabolic one. So we all know that obesity increases the risk of cardiometabolic diseases consisting of coronary artery disease, stroke, and type 2 diabetes, but we don't actually know how. And together with colleagues, I hypothesized that circulating protein may mediate this relationship. Why? Because circulating proteins are strongly influenced by obesity, and they can be easily measured and often moderated, and many drugs target circulating proteins. But as you know, they are involved in super complex biological systems, so observational analysis may be super biased or confounded. So the aim of this study is to identify circulating protein mediator with causality. So we designed a two-step approach. In step one, we identify the effect of BMI on plasma protein using a genetic epidemiology method called Mendelian randomization. How many of you guys are familiar with MR? I guess Guillaume did a presentation here. Okay, good. Okay, so but I'm going to cover this again in the next slide. So yeah, so this is a method which uses GWAS summary statistics to make a causal inference. For this, we use BMI GWAS of 700,000 individuals from UKB plus Giant as an exposure, and we use 4,907 plasma protein level and 35,000 individuals and from the decode cohort. Both are publicly available data, so you can run this on your own computer. Maybe it takes a bit of time, but you can still run it. Okay, so after identifying circulating protein influenced by BMI, we move on to step two. So in step two, we identify the effect of BMI-driven proteins on cardiometabolic diseases, which include coronary artery disease, ischemic stroke, and cardioembolic stroke, and type 2 diabetes. So coming back to what's MR. So MR is a genetic epidemiology method, which makes causal inference, and is often compared to randomized control trial. So RCT, or randomized control trial, is the best way to make causal inference. If you like to test whether protein A is causal for coronary artery disease, the best way to design like 1,000 individuals hundreds of thousands of individuals RCT, but it's cost so much money. It's going to cost 3 years, and it's not really easy. But you can use MR instead. So MR take advantage of random allocation of allele. So in RCT, you randomly allocate a drug such that confounder would be like age, sex, that kind of factor would be equal among the case group and control group. So that's why you can test the effect of drug on outcome, which we see. But here, thankfully, the nature decided to allocate your allele randomly. This is called Mendel's second law. So we can take advantage of this. There is an allele which increase protein A stable, and there's an allele which doesn't increase protein A stable. So we take a closer look at these two groups, people with effect allele, people without effect allele. So it's really like people who are assigned to be on drug from the very beginning of their life versus for now. This way, the confounders are being equal between the two groups, and we can compare the outcome. This means that we can use genetic variants as an instrument to test the effect of protein A on coronary artery disease. And this is how you actually do that. So you go to protein A's GWAS. The x-axis is a given variant effect on protein A. So there is a variance which increases proteins A slightly, like this. But there is a variant which increases proteins A's level massively, like this. And you plot these variants beta on the outcomes. You go to CAD GWAS, check this variant. What's a beta for this variant in CAD GWAS? Okay, so it turned out this variant, which increased protein A level slightly, also increased the risk of coronary artery disease slightly. And when you take a look at this variant, which increased protein A level massively, increased coronary artery disease massively. So you keep plotting this thing and scatter plot, you draw a regression line, and this slope is a causal estimate. If there is no effect, you will find no slope. You just see like a random dots here. But in this case, you clearly see a positive slope here, which means that protein A has a causal effect on coronary artery disease. And you're seeing this through genetic variants. Any questions so far? Great. Okay, so this is step one. We tested the effect of BMI on circulating proteins. We know that BMI have a massive effect on circulating proteins, but we would like to understand this in the causal context. So we tested 4,907 proteins. So this is doing 4,907 MR. And after MR and heterogeneity, directional polarity, reverse causation tests, which are kind of sanity check, we identified a bunch of proteins which are influenced by coronary artery, BMI. And this is a volcano plot. X-axis represent beta from MR, y-axis represent minus log 10 p-value from the MR result. So for example, leptin, which is a well-known protein increased by obesity, actually has positive beta and very strong p-value. This means that increased BMI leads to increased plasma protein level of leptin. We also know that increased BMI leads to chronic inflammation in your body. And we do see a marker of chronic inflammation, which is CRP here. So a bunch of them are known, but there are a good number of guys who are not known. So we're interested in them. But these positive controls serve as a kind of sanity check. But there are some arguments that BMI is not the best proxy for obesity because it's just use height and weight. But we know that you can have the same BMI even if you're super muscular or not. So that's why we also use body fat percentage in the same flamer. We tested the causal effect of body fat percentage and the present protein. And this is MR beta. This is MR beta for the same protein. You see an excellent concordance. So this actually tells that BMI on the population level, it's a pretty good proxy for obesity. Okay, so now we have identified a good number of proteins which are influenced by BMI and body fat percentage. So let's move on to step two. In step two, we identify the effect of these proteins on cardiometabolic outcomes. So same thing, 2 sample MR, and this time we are using a genetic variant called protein quantitative trait loci PQTL as a proxy. So what is this? So it's a variant that controls plasma protein level. So the more G genotype you have, the more G allele you have increased plasma protein level. So something like this. And after MR sensitivity analysis, colocalization, and mediation analysis, which are non-genetic epidemiology method, which I'm not going to cover here, we identified 8 protein disease associations. So for CAD, it looks like this. The top fits for a protein called COL6A3. So you can interpret this such that SD increase in COL6A3 increased the odds of coronary artery disease by 1.47, and you also see PCSK9, which is a super well-known target, and there's already a drug which target PCSK9. It's called PCSK9 inhibitors, and it works extremely well. It's gonna drop your LDL by 90% or something, and it's gonna decrease your risk of coronary artery disease. So we know that this works, and we nicely capture this. And so this is a protein hits for ischemic stroke, cardioembolic stroke. F11 belongs to a coagulation pathway, so it makes a lot of sense. And we also have 3 hits for type 2 diabetes. So it's nice to identify a protein that lies between obesity and cardiometabolic outcome, but it would be nicer if we can modulate that. So to ask that question, we perform multi-variable MR to see what will be the like a finer picture of this. So we tested the independent effect of body fat mass and fat-free mass in the multi-variable framework and see during what's the independent causal effect of fat mass on plasma protein mediators and what's the independent causal effect of fat-free mass on them. And it's really interesting to see that COL6A3, F11, and these guys increase plasma protein, SD increase in fat mass increase these plasma protein level, whereas increased fat-free mass, which are mainly muscle, leads to decrease level of plasma protein level. So we intuitively know that like more fat is bad and more muscle is good. And this kind of agree with that understanding. So human bodies are really fascinating, and genetics is the one way to disentangle it. Okay, so I don't have time to go into the detail of all proteins. So let's take a look at the top guy, COL6A3. So what is COL6A3? So COL6A3 is a protein called collagen type 6 alpha 3, and it's expressed in many human tissues, including adipose tissues, artery, and fibroblasts. And what's really interesting about this protein, this C-terminal is cleaved into another protein called endotrophin. And there's a mice study saying that this endotrophin is associated with fibrillar inflammation, fibrosis and inflammation, and insulin resistance, which is basically the systematic chronic inflammation in your body, which leads to cancer, coronary arteries, and a bunch of bad things. And they did this experiments in some mice obese adipose tissue, and they also found that by inhibiting this endotrophin, they can improve this adverse syndromes. But this is a mice study, which do not necessarily translate into humans findings. So there is a cross-sectional human studies as well. So in the cross-sectional studies, endotrophin was associated with cardiovascular event, which is good. But cross-sectional study cannot disentangle between the causation and consequence. So there is a famous study targeting HDL to treat CAD, and it massively failed. So they invested tens of thousands of money. Phase 3 trial is published in NEJM, and there was no effect. But if they have done MR, we have already known that lifetime exposure to the reduced HDL does not lead to increased coronary artery disease. So that's a kind of classic example where MR could have saved tons of money. So that study was actually based on observational study and mice studies. They saw it works, but it didn't. So it's really important to understand what's causal. And with this study, we use human genetics to show that endotrophin is causal for coronary artery disease. Of course, based on some MR assumptions, which we try to minimize bias using multiple sensitivity analysis. Okay, so what's more interesting is there is to analyze targeting N-terminal co663 and C-terminal co663. So we are using a protein measurement platform called SomaScan, and they design and analyze such that some analyze targeting same protein. But this was a complete coincidence. They didn't like intentionally target N-terminal, C-terminal this, but I communicated with SomaLogic people, and it turned out one target N-terminal, another target C-terminal. And it was so nice to see that only C-terminal co663 increased the risk of coronary artery disease, whereas N-terminal was completely known, which completely agree with the prior biological knowledge that only C-terminal got cleaved and go into the circulation. But there are some bias to any platform. So that's why we decided to use another platform called Olink Expo assay. Actually, not we, like a UK biobank already use Olink Expo assay, which is another antibody-based protein measurement technology to generate a PQT of co663. So we use that variant to test the causal effect of C-terminal on coronary artery disease, and we nicely replicated the causal effect. So different platform and different cohort pointing to the same causality, which is really nice. And we also performed Cox regression survival analysis in the UK biobank. And the more co663 level you have at baseline, the more coronary artery disease you have in 10 years. So observation analysis also supported it. And when you take a closer look at the genetic variant you're using to proxy the co663 level, so it lies in the active enhancer region, and the adipose tissues, coronary arteries, aorta, thoracic artery, tibial arteries. So and this disrupts the DNA binding too. So this makes complete sense because this variant had negative beta in co663 GWAS. So it looks like this variant affects the genetic bindings, so DNA binding. And this actually leads to the decreased co663 expression, which I found in GTEx. And then it leads to reduced plasma protein level. So everything lies up. And to further understand the biological role of co663, we perform a single cell RNA-seq analysis. And in adipose tissue and coronary artery disease, which are two relevant tissues. And we found that co663 is highly expressed in adipose progenitor stem cells and fibroblasts of coronary arteries. So fibroblasts are kind of easier to understand. So it's causes fibrosis, probably in coronary artery disease, and do bad things. So more fibrosis, more co663 expression, more cleavage, more endotrophin, do bad things locally. Okay, which makes sense. Adipose tissues, which looks a bit unclear, but actually it's not. So adipose progenitor stem cells is kind of stem cells for mature adipose tissues and bone. And so when I performed PWAS, phenome-wide association study using this variant as a proxy to co663, I found that reducing co663 leads to increased like a bone mineral density. So by reducing co663, it looks like your CAD risk is reduced and your bone gets thicker. Why? It looks like this kind of answer that. This adipose progenitor stem cells can go into mature adipose tissues and bone. By inhibiting this pathway, ADPC going to mature adipose tissue, this guys go to more bone and your bone get thicker. So looks like PWAS and this single cell RNA-seq thing show that you can target co663 safely, and also you may have a good side effect. It's not a side effect, but good effect on bone as well. We also checked whether BMI influence whether C-terminal and N-terminal differentiate. And we found that BMI influenced both terminal, but it preferentially increased C-terminal with no confidence overlap. So taken together, more obesity, more caustic history, more cleavage, more androtrophine. This part is probably, but it's likely. And more androtrophine leads to increased risk of coronary artery disease. And yeah, so this is pretty much it for the first section. Overall summary, proteome-wide MR can identify certain protein mediator. And domain-aware MR, this, to my best understanding, this is the first study which actually used domain-specific causal inference to disentangle the biology. And we use multiple layers of evidence to strengthen the evidence because every method, every cohort, every technology have bias. So it's always nice to triangulate the evidence. And if the more layers point to the same thing, the stronger the evidence is. Okay, any questions so far? Good. All right, lots of time. Okay, it's 30 minutes. Okay, good. So now let's move on to the multi-ancestry atlas of protein disease association. So why do we have to do this? So you might have heard that GWAS study have a hugely bias to Europeans. So that y-axis is the number of GWAS, and the x-axis is the year published. You can see like most of them are Europeans. This is getting a bit better, but still this is the case in this era. And MR relies on GWAS. So apparently this applies to MR as well. But if you keep doing this, this may lead to a disproportionate effect on the healthcare, because at the end of the day, you're interested in using these human genetics data to do drug discovery or precision medicine. But if you only study European ancestry individuals, your treatment strategy only applies for Europeans. So that's why we have to be inclusive. And so that's why we decided to do multi-ancestry proteome-wide MR to get a comprehensive picture of protein dissociation and identify potential therapeutic targets. So this is a massive collaboration, which is led by Chen-Yang. Thank you so much, Chen-Yang. And with Tianyuan. So 3 of us decided to work on this a year ago. So I have been working on this multi-ancestry BMI thing, but I had too much to do. I didn't have time, and it was kind of lying on my cluster. But Chen-Yang had free time, so he decided to, okay, why don't we do this? And Tianyuan supported me. So this is a group collaboration between QLS and Adrian, who is a postdoc at University of Lausanne, and Kyoto Universities, who provided PQTO data in East Asian, and Jason Flanick, who is my previous mentor at the Broad Institute, and Vincent, who is at CERC, so many other people. And this is a kind of advertisement, but Chen-Yang will give an oral presentation at ASHG and IGF. So giving an oral presentation at ASHG is such an honor. It's an honor for top 6%, and he's also shortlisted for Williams Award, which he's gonna compete at the conference. So this kind of highlights the importance of this project. I'm very proud he got this award. Okay, so let's go. So this is a study design. So we are trying to create a multi-ancestry atlas. How are we gonna do that? So we have your 3 ancestry, European, African, East Asian, 3 ancestry, 7 cohorts. We try to use as many cohorts as possible because there are some bias to specific bias to each cohort. And we're gonna test up to 4,907 circulating proteins on 355 unique outcomes. So how did we create this outcome? So we went to GWAS catalog like Nature Genetics or anything, and we kind of manually created this. It was such a back and forth, like such a hard effort, but thanks to Chen-Yang's work, we were able to do this. Yeah, okay, so this is the details of the 4th cohort. We are using ARIC decode, Finland, UK biobank for Europeans, and ARIC and UK biobank for Africans, and East Asian data from Kyoto University. This is not published yet. I'm a contributor to this study, so that's why I have access to SomaScan base data of plasma protein measurement for 1,823 plasma protein in, no, sorry, 4,300 plasma protein in 1,800 individuals. Okay, so this is the unique number of protein tested. So of course, you can test more proteins in Europeans, but you can also test 581 plasma proteins in East Asian. You can test 1,146 plasma proteins in Africans. And what's really important is there are some proteins which are only able to be tested in non-Europeans. So there are 17 plasma protein which will only be able to be tested in East Asian, and there are 119 protein which are able to be tested in Africans. So why? So this is one of the reasons. So when you take a look at allele frequency of these variants, so just to remind you, we are using genetic variant to proxy the plasma protein level. So when we take a look at these variants proxy, these variants have a good allele frequency, like a pretty common in African, but they are super rare. A good number of them are super rare in Europeans. So around 70% of them are lying here, like with allele frequency of virtually 0 in Europeans. So if you like to instrument these proteins in European data, you're gonna need tens of thousands of data, and which is not gonna happen because it's quite expensive to measure plasma protein. So current Olink platform costs around 1,000 USD. So if you do this for 100,000 visual, you're fried. I'll try. So I'm gonna do it. So, so that this really showcased the power of multi-ancestry inclusion. So by including non-European data, you can now instrument more proteins, and you can test the biology causality of these proteins on many outcomes. And we suspect that these results, causal and inference results, can be applied to other ancestry. It's very hard to believe that some important proteins are only affected in one ancestry. It's more likely there could be some protein like that, but I assume it's more like the case that these proteins can be only instrumented in Africans, but these biological functions can be applied to others. So multi-ancestry inclusion will not only be beneficial for these underrepresented minorities, but be helpful for everyone. Okay, so we tested up to 726,000 plasma protein outcomes here in European, African, and East Asians. So we identified a massive number of proteins, and apparently I don't have time to go into each of them. So this is a cardiovascular network plot. This is a autoantibody network plot, and so forth. But let me take a deep dive into one disease, which is called inflammatory bowel disease, because I covered cardiometabolic disease and the previous section, so I'm gonna this one. So it's a heart disease. So I have seen patients with IBD, and he was 19 years old, but he had to resect like a majority of the colon because of this disease. So this causes inflammation. Sorry. So this causes inflammation in your body, in your intestine, and the inflammation is so severe that they will kind of stick to each other, and your intestine will be closed. So you can't eat anything anymore. How we treat that, we kind of cut that section and connect it together. Now it's open again. But apparently that's very hard. And there is a bunch of anti-inflammatory disease, but none of them are effective enough to cure them. So there are more to be done about this disease. So we took a closer look at which proteins are causal for inflammatory bowel disease, which is highlighted here. So let me explain how to read this network. So there are plasma protein here connected to inflammatory bowel disease. The blue line means negative beta. So the increased level of this plasma protein leads to increased risk of IBD. And red line represents positive beta, so more protein, more risk of IBD. And the line, the thickness of the line represents the number of the cohorts supporting it. So the thicker the line is, the more robust the associations are. And you can see a bunch of protein connected to IBD. So these are the core proteins. And one of them is IL-12B. So it has a red line connecting to IBD, okay, so it's bad. And it's also have a red line connecting to Crohn's disease. So our MR is saying that increased level of IL-12B leads to increased Crohn's disease risk. And actually it's already known, or there is even a drug which are FDA approved. So this drug is already used as a active phase and chronic phase treatment for coronary artery disease. So we're replicating that findings. It looks like there is another line going to ulcerative colitis, good, but actually this is already known. So this drug is also from ulcerative colitis, which is another IBD. So IBD consists of Crohn's disease and ulcerative colitis. And this targets are causal for both, and they are already drugged. And there is also another line connecting IL-12B to psoriasis, another autoimmune disease. And yes, there is a drug for that. So this IL-12B is a very nice target, which is like we target this protein and you can hit 3 outcomes. So it's an amazing protein. So it's nice to kind of replicate these findings, but we're interested in new findings as well. So that's why we took a closer look at this network and identified one protein which are connected to multiple diseases, which is IL-1, RL1. So what is this guy? So this guy increase inflammatory bowel disease, Crohn's disease, ulcerative colitis across multiple cohorts, across multiple ancestries. And when we do a follow-up analysis in the UK biobank, it's increased baseline IL-1 leads to increased 10-year risk of IBD, Crohn's disease, and ulcerative colitis. So observation analysis support that. And when we do gene expression analysis and the bulk level, we found that Crohn's disease patients, ulcerative colitis disease patients have increased IL-1, RL1 expression in ileum, colon, and rectum. So it looks like more, if you have this disease, more IL-1, RL1 expression in the disease area as opposed to circulation to biobank. And when you take a look at the single cell resolution, it's expressed in mast cell, which plays a key role in immune system. And it's actually known that IL-1, RL1 is specific to mast cells or helper T cells. So this kind of agrees with the previous findings. So nice to identify some target, but can we do something about this? The answer is yes. So there is actually a drug in development targeting IL-1, RL1. So this drug is now in phase 2, targeting atopic eczema, atopic dermatitis, and another lung disease called COPD. So this kind of indicates that, yes, this drug is there, but it's not tested against IBD or Crohn's disease or ulcerative colitis. But actually this is developed by Roche, and we are communicating with Roche, and maybe we could suggest that, hey, this drug can be effective for IBD, so why don't we test that? So we did extensive preparation using 3 druggability database, one from Finnetrol, another from drug bank, and another from Open Target. And we created like an extensive map of druggability plot for the identified protein, which I do not have time to go into the details, but this paper will be hopefully up in the archive pretty soon. Okay, so I had 15 minutes. Any questions so far? Good. Okay, so let's move on to the last topic. So I'm gonna cover the new multi-ancestry multi-omics biobank called Bioportal and disentangling the heterogeneity of type 2 diabetes. So what's Bioportal? The bioportal is a new multi-ancestry, multi-omics biobank established at Jewish General Hospital, a teaching hospital of McGill University. And we're gonna recruit 12,500 individuals with diabetes over the course of 5 years. We already secured $9.4 million in CAD from public and private entities such as Jewish General Hospital Foundation, McGill Research Program, CQTM, and Rush. And we will do genetics analysis called blended genome exome sequencing, which consists of whole exome sequencing with 30x, plus whole genome sequence plus imputation for everyone. And we're also gonna do proteomics with Olink HT, which can cover 5,400 proteins for 3,000 individuals. And we are doing a deep phenotyping, including body compositions and fasting instincts and so forth. And we're connected to the EHR record. And the key advantage of this study is a strong diversity. We already recruited 2,200 individuals, and more than half of the participants are non-Europeans. So there is no cardiometabolic biobank with this scale, with this diversity, with this deep multi-omics data set. So I'm envisioning a very strong future for this biobank. But why do we need this for diabetes research? So the answer is diabetes are killing people. So there are 1.5 million people dying because of diabetes and its complication every year. I can't really believe this is still happening in 2024, right? But actually this number is kind of increasing. And this is a landmark paper published in 2015 about the amazing drug called empagliflozin, which reduced the cardiovascular risk by 14%. So this study has been cited like 20,000 times. So some doctors said this is the second greatest discovery after the discovery of insulin, because we try so hard to tackle the cardiovascular deaths in diabetes patients, but we haven't been successful until 2015. So this is amazing. But when you look at the number, it's just 14%, and there's a good number of people who are still experiencing the cardiovascular events and get killed. So there are more work to be done. And you might wonder why, why don't we just lower the glucose level? So this is one study which I did with a junior endocrinology resident back in Japan. And we show that there is a dose-dependent association between glucose control and cardiovascular risk. So HbA1c, the higher it is, the bad it is. So it's bad. And so you see that there's a negative flow, which means that the better glucose control leads to better cardiovascular risk. But it's good, but you see there is a huge substantial heterogeneity here. So what are they? So apparently glucose doesn't explain everything. So you might have been thinking diabetes is just a disease of glucose, but actually it's not. It's a more complicated disease. So now recently it's increasingly being recognized that type 2 diabetes is not a single disease. It's a collection of metabolic dysfunctions. So there is a study in Lancet Diabetes Endocrinology using clustering algorithm in very simple clinical variable: age at diagnosis, BMI, autoantibody, HbA1c, HOMA2 beta, which is the proxy for insulin secretion, and HOMA-IR, which is an insulin resistance index, which you just have to calculate this, you just have to use fasting insulin, fasting glucose. And so these are like routinely measured variable in clinic. So they just did simple k-means clustering with these variable, and they identified 5 clusters. And some clusters has like low BMI, other clusters have high BMI. But why this is relevant? Because it is associated with disease progression. So insulin resistance group, which have high BMI, high insulin, high glucose, have increased risk of end-stage kidney disease and coronary artery disease, whereas insulin deficient group, who are typically lean, which means don't have high BMI, but have high glucose level, have increased risk of diabetic eye disease. So we are currently treating them as one single disease. You're thinking this is a type 2 diabetes, let's just lower glucose. But that's apparently not enough. There is a heterogeneity underneath, which explains the disease progressions, and there is definitely an underlying biology. And disentangling the heterogeneity type 2 diabetes has been such a hot topic recently. This is GWAS study trying to disentangling that. This is another GWAS study trying to disentangle that. And this is a clinical study which are trying to disentangle that. So there are two approaches, genetics and clinical. But clinical variables are noisy, apparently. If I have flu, like my measurement would get messed up. But genetics will not change even if I have flu. So genetics are more stable. And as a human genetics, I'm much more interested in using genetics to disentangle the heterogeneity of type 2 diabetes. But the problem is existing large scale biobanks are not good enough. UK Biobank has large omics data, good genetics data, but it's missing clinical variables. It's missing key clinical variables such as duration of diabetes. We don't have insulin level. We don't have autoantibody level. So we can't really exclude type 1 diabetes, which is the autoimmune diabetes. And we don't know how long this patient had type 1 diabetes, type 2 diabetes. So yes, it's a good resource, but it's not nice. It's not the best resource to do diabetes research or any type of disease research because it's a disease agnostic biobank. So this is why we need bioportal. So we will use large-scale omics data and deep clinical phenotype data in the bioportal to disentangle the heterogeneity of type 2 diabetes with characterization of each cluster and cluster-specific drug discovery. And we're also now working on a new Bayesian approach to identify putatively causal genes for each diabetes subtype by generating full GWAS of this. I was hoping to discuss this, so I just realized this is a public seminar, which will be posted on YouTube. So I decided not to do this because this is ongoing work and will be published soon. So I'm very much looking forward to share more details about this in the near future. But this is a great collaboration with Dr. Jason Flank and Dr. Jose Flores and many others at the Broad Institute of MIT Harvard. Okay, so I also like to illustrate one usage of this type of biobank. So this biobank can recall patients by genotype or phenotype, but why is it good? So let's learn from an amazing success story of the human genetics. So PCSK9 is a good drug target, and it has been drugged, and it has been super successful to lower LDL cholesterol and protect against coronary artery disease. So this actually disease or like variant were identified in French Canadian population. So French Canadian populations are kind of bottlenecked because of the founder effect. So they have an enrichment of unique variants. And there were families who had loss of function variants in PCSK9 living in Quebec. And their LDL level was like super low. And when they take a closer look, their coronary artery disease was super low as well. So inspired the findings, people thought that, okay, maybe PCSK9 could be a good drug target. So let's collect people with loss of function PCSK9 variants. So this is a well-known PCSK9 loss of function variant, and this is the LDL level without that PCSK9 loss of function variant. So this is a people with that PCSK9 loss of function variant. So you can see LDL cholesterol histogram is shifted towards the left, which means if you have this PCSK9 loss of function variant, you have a lower PCSK9 level, you have lower LDL cholesterol level, and lower coronary artery risk. And there was another study using another variant showing the same thing. So even if you change the genetic variant, you see the same thing. Lower PCSK9 level, lower coronary artery. So this just tells you that you're using genetics variant as a proxy to PCSK9 level. And you're seeing the effect of lower PCSK9 level on coronary artery disease. But this is a kind of coincidence, but we can do this in a systematic manner. So we have a target of interest, such as endotrophin, which we identify through the biobank scale analysis. We can recall patients based on their genotype. We can recall patients who are with and without genetic variant, which lowers or increase PCSK9 level, and do deep phenotyping and design a study. So this will help you save a massive amount of money, because if your drug doesn't lower endotrophin level in the people who have high endotrophin level, it will not gonna work in the general population. So this will help you save massive amount of money. You don't have to spend like $890,000. No, I think $890 million. Yeah. So this kind of recall by genotype studies is really helpful, and it has been like empowering many studies, but unfortunately, not many biobank has done this, so we would like to do this. And the last thing, as a clinician, I'm interested in precision medicine. So polygenic risk score, I guess some lecture kind of covered polygenic risk score. It just aggregate your genetic variant to see your genetic burden or individual person genetic burdens on a particular disease. And this is a PRS score of coronary artery disease on x-axis, and y-axis is the prevalence of coronary artery disease. And you can see that if you happen to belong to the top percentile of CD polygenic risk score, your CD risk is more than 5-fold higher than the people with the lowest polygenic risk score. This is equivalent to monogenic form of disease, such as familial hypercholesterolemia. We are treating familial hypercholesterolemia in clinics. We prescribe PCSK9 inhibitors. We prescribe the strongest level of statin. So we act upon it because we know that if we don't, people will die. But we're not doing anything about these people yet. So we would like to act upon polygenic risk score and other genetic architecture in this biobank. So in summary, I hope I have, to some extent, convinced you that we can leverage human genetics for drug target discovery with an ultimate goal of better clinical care. And our goal, our lab's goal, is to human genetics for drug target discovery and better clinical care. And our approach is to ask clinically relevant questions and address them with human genetics. So we gonna use additional resources such as UK Biobank, all of us, which is 207 individuals, Biobank led by NIH. We have access to the Mass General Proteome Biobank dataset because I'm also affiliated with abroad. We have access to Kyoto University's proteomic dataset, and our lab is contributing to GOS consortia, including type 2 diabetes global genomics initiative, MAGIC consortium, which are glycemic group, TRAGE consortium, and coronary artery disease, anthropometric rate, and osteoarthritis. But this is really to leverage human genetics to identify drug targets and include clinical care. So thank you so much. I'd like to thank the amazing lab member, Chen-Yang Su, and the CERC member, including Vincent, Cirui, my amazing mentor, Ben Richards, and amazing collaborator, Ken Yuan Lu, Jason Flanick, and many others. And thank you for your attention. Happy to take any questions. All right, so thank you so much.
I have a question for you.
Sure.
When you mentioned the clustering of type 2 diabetes
Yeah.
into 6 different subtypes. I know that these are never very clean clusters.
Yeah, that's
Um,
clinical variable based.
Yeah. But, can you? How clearly do they separate genetically?
Yeah, so that's an excellent question, and that is done by these two papers. So
Uh-huh.
I do have a slide for that. Okay, let me. Okay, to first answer your question, actually, it's nicely agree with each other. So phenotypic clustering, genetic clustering, they point to the similar clusters, which is very nice and very convincing. Of course, it doesn't completely overlap, but the key cluster do.
Okay.
Yeah, so this is how you do with genetics. So you take genome-wide significance type 2D signal. So you take a type 2D genome summary statistics, pick the genome-wide significant independent one, and that's variant 1, variant 2, variant 3. And you also take the diabetes associated traits such as fasting glucose, BMI, and triglyceride. The selections are arbitrary, but 37 cardiometabolic traits were used in the Nature paper from type 2 DGI, and Miriam Elder, my collaborator at the Broad, used 110 traits. And you get the Z-score of corresponding variants in corresponding traits, and you fill the cell. So this is how you get this gigantic matrix,
Yeah.
and you apply clustering.
Yeah.
So Nature paper apply k-means, and Miriam in Nature Medicine paper apply non-negative Bayesian metrics factorization, which is
Yeah.
a soft clustering method.
Yeah.
But actually, it's produced pretty similar results, which is also nice because different methods, same results.
Mhm.
And this is how the cluster looks like. The one cluster looks like this, another cluster looks like this. We have to give names, so that's why I decided to call this a beta cell cluster, protein cluster, obesity cluster, lipodystrophy cluster. And when you take a look at the Z-score, so this is a 0. So this cluster have very strong negative Z-score for beta cell cluster. Oh, sorry, the HOMA beta. HOMA beta is an insulin secretion function. So it has a very negative beta, and it also has a very positive beta for proinsulin, which is a precursor of insulin. It's known that if you have type 2 diabetes, you have to secrete a lot of insulin, you have to produce a lot of insulin, so your pancreas works very hard, and you start to produce immature insulin. So that's why in clinic, we do see increased proinsulin level in early type 2 diabetes patients, and we do see positive beta in this genetic cluster as well. So this pattern agree with what we know as a beta cell dysfunction, pancreatic beta cell dysfunction. That's why they call it beta cell cluster. And another thing is obesity cluster, which doesn't have the signature with the insulin secretion thing, but insulin resistance. This is how well insulin work in your body, and this is basically proportional to your obesity level. And this is a very strong beta for BMI, waist circumference, fasting insulin. This really mimics the type 2D patient's picture with obesity.
Okay.
And another is lipodystrophy. The pattern are pretty similar to obesity, but it has negative beta for BMI. So this means that this person is lean, but have a very bad metabolic signature. And we do see patients like this in clinic. And actually, the previous Lancet Diabetes and Endocrinology paper identified this group, obesity group, lipodystrophy group. They name it differently.
Yeah.
They call severe insulin deficiency diabetes.
Yeah.
Sorry, insulin deficient diabetes is this one, and severe insulin resistance diabetes. Third is this group. So it's named differently, but this is a very similar picture. Yeah. Go ahead.
I had a question on, you mentioned having ancestry specific proteins.
Yeah.
Were those from SomaScan or were those from Boeing?
So there are, okay, so we use both platforms and they are SomaScan because, like we only have SomaScan data for East Asian.
Okay.
And for African, so for the sequence comparison, we have to do that.
Okay, and then so when you say uniquely.
Oh, but sorry, no. So that's compressed into protein coding genes. So
Okay.
actually there are some protein which are instrumented by Olink platform. But that's only in Europeans and African ancestry for which we have the data. Yeah. The East Asian data is small scan.
Thanks.
Yeah, sure. Yeah, like we are, this, I'm gonna cut it here, but so this individual level data is the Asian individual level is very valuable. So it's only Chinese biobank, which also had like a large scale data, but it's an O-link and it's covered fewer proteins. So yeah, I see a huge opportunity in this, and that's why, Chen and I are working very hard on this multi-omics biobanking, and we are trying to do more with these data, but we don't have many people, so you're always welcome to join us. Okay, bye.
I have a question about so you applying MR, and in this new cohort that you're building with the US hospital, you're going to have a proportion of Latin samples. So
Yeah.
What's the challenge of using MR without mixed populations? Because you have the multi-ancestry one, but you don't have one for example, the Native American component.
Yeah,
And also when you have a mixed samples, that you're going to have a lot of like complex interactions there because like a mosaic of a lot of different continental sources. So how do you address that?
that's an excellent point. That's a great question, and we can't. We should do MR in a ancestry stratified manner because of the reason you mentioned. And we will not have power to do that with this cohort. So this cohort shouldn't be considered as a source of MR. The two sample MR, we can do that in the large scale biobank. We identify target, but we have to do a follow-up analysis on that. How can we do that? So we identify neutrophin, and that's going to be published in Nature Genetics, but I'm happy, but I will be even happier if I can make a drug for this. But what will be the next step? We would like to recruit people with higher neutrophin level in this cohort based on their phenotype or genotype, and do screening or like MRI study, how the plaque flowing in their coronary arteries and so on and so forth. So we can do further studies and build the data and act upon it. Another way to use this biobank will be like clustering, right? So we can do clustering based on proteomics data, genetics data, clinical data, aggregate them, cluster people, and identify signatures, and then go to large scale biobank to test if like people with similar signatures have like increased risk for coronary artery. So there's a kind of like two ways to use this data. But as you mentioned, in terms of MR, no, we shouldn't do this. Right.
Thank you.
Thank you so much.